Huntington's disease is a neurodegenerative disease that causes breakdown of nerves. It deteriorates a person's mental and physical abilities, causing a lack of coordination, chorea, and problems with mood. You can see the difference in a normal brain versus a brain with Huntington's. Neuronal cell death is most prominent in the basal ganglia of the brain, where there is an expansion of the lateral ventricles.
We started seeing signs and symptoms of Huntington's disease in my dad when he was 50 years old.
I am 1 in 200,000 people with Huntington's disease.
The Huntington gene is found on chromosome 4 and contains a triplet repeat on the N terminus. CAG is repeated 10 to 35 times in a normal person, but more than 36 times in someone with Huntington's disease. CAG codes for glutamine, so people with Huntington's will have 36 or more glutamines in a row. A lot is still left unknown about Huntington's disease, such as why extra glutamines cause mutated HTT genes. What we do know is that the mutated protein aggregates within the neuronal cells of the caudate and putamen of the basal ganglia, causing neuronal cell death.
This is the non-mutated Huntington protein with 10 to 35 CAG repeats. HTT is largely alpha helical and consists of three major domains: the N-terminus, C-terminus, and heat repeats. Huntington contains heat repeats, which are repeated sequences of around 40 amino acids believed to be involved in proteins transport, microtubule dynamics, and chromosomal segregation.
I found out that I had a repeat number of 42, which identified as carrying the gene of Huntington's. I will develop Huntington's around the age of 42. But since my father carried the gene, that deducts 5 to 6 years off of that. So I will start showing signs and symptoms around the age of 35, 36.
Cellular function remains unclear because cryo-EM has been unable to capture photographs of the protein due to its large floppy shape. The blue section represents Mary's 42 CAG repeats. However, the exact structure of this mutation is still unknown.
Brain-derived neurotrophic factor is an important factor in HD. Huntington specifically enhances vesicular transport of BDNF along microtubules. Since the function of HTT is unknown, we can hypothesize that Huntington promotes BDNF transport along actin and myosin. The loss of this function might contribute to HD.
Fragments at the N-terminus of the mutant Huntington form due to caspase cleaving or abnormal splicing of Huntington mRNA. These fragments contain long glutamine tracks, which cause them to clump together through hydrogen bonding. Because mutant Huntington contains a mutated nuclear export signal, the N-terminus Huntington fragments are trapped inside the nucleus, disrupting transcription of other proteins and other nuclear functions.
MHTT negatively impacts cells through the formation of neuronal inclusions, which are aggregates and whole Huntington proteins. These inclusions mainly accumulate in axons and dendrites, where they damage cell structure and warp the nuclear envelope. They also inhibit the transport of important molecules and structures, such as mitochondria, between the cell body and the axons and dendrites. This causes mitochondria to clump in the cell body, creating more complications. MHTT damages the ubiquitin protease system. Normally, when a cell identifies a misfolded protein, the system tags the protein with ubiquitin. Proteases then destroy these ubiquitinated proteins.
My father is no longer able to drive, no longer able to care for himself, no longer able to do his hobbies like hunting and fishing. He lives at home with 24-hour care, and it's changed our lives a lot. Knowing that I have this life-altering disease has been scary, but it changes my perspective on life, and I live a happier, more optimistic life.